Oh, the first question is which of the following is found in all prokaryotic cells, but only some eukaryotic cells, which is the cell wall, because all prokaryotic cells have a peptidoglycan cell wall, but only some eukaryotic cells, such as the plant cell and the fungi cell, have a cell wall.
Yes, and for the next.
Viroids,
Next question is okay for you?
I think so. Viroids are infective agents with protein-free with low molecular weight RNA. Is an RNA molecule. I think that most people, yeah.
Okay, so viruses, they are infective agents in plants. Okay, so we find some plant disease where what the problem, sorry?
No.
So we find some disease in plants were affected by this virus, and it's composed from only single-strand RNAs without proteins. Okay, and for the question number 3.
The section number 3 is the false statement is they are sensitive to antibiotics because only the prokaryotic species are sensitive to antibiotics. Viruses, I don't think that it isn't easily sensitive to this kind of treatment.
Okay, so, all right. For the next question, I see that you are the second winners in these games, and you have the wrong answer in these 2 questions, but you explained very well. The question number 4, is this okay for you?
Yeah. The cell wall of gram-negative bacteria are thicker than gram-positive bacteria. Is that the correct answer? I think it must be the A, right?
Yes.
Gram-positive, yeah, because the gram-negative has only one, thinner cell wall between 2 membrane, and the positive gram-positive bacteria have a thicker cell wall, which has only include one membrane.
One layers, but very thick layers of peptidoglycan.
Yeah.
And gram-negative, they have a 2 layer, but only 1 layers of the peptidoglycans and 1 outer layer.
Yeah.
And these 2 thick layers make them is more dangerous, because if you're affected with some disease by the gram-negative, is more difficult to treat. And for the question number 5.
The question number 5 is the cell membrane structure, because the cell membrane structure of archaea bacteria are more alike to the eukaryotic cells, but not like the eukaryotic. Okay.
So if not correct for these questions, I think the cell membranes of archaeobacteria, they are quite unique for the domains. They different from eubacteria, and also they different from the eukaryotic cells. Just because the archaeobacteria have one cell membrane with 3 characteristics. First, the linkage between the lipids is ethers, but not ester, like in eukaryotics and also in bacteria. And the second characteristic is they the side chains the side chains of archaeobacterias is different, and the third characteristics is they are one phospholipid layers. But for the eukaryotic cells and bacterial cells, they are bilayers. But in archaeas, they fuse together to make only one layer. So by that three characteristics, the cell membranes of archaeobacteria is become more rigid, and they can live in very harsh environments, and they can become the extremophiles organism. So for the question number 6, who volunteered to explain these questions? Or if no, I will. So mechanism action of penicillins is?
I think it's cell wall synthesis inhibition.
Yes, cell wall synthesis inhibition. So in the previous lesson, I only required you to remember for me just 5 antibiotics, right? So in the mechanism of action, if they inhibit the cell wall synthesis, we have the penicillins. If they inhibit the nucleic acid synthesis, we have quinolones and ripamycins. And if they inhibit the protein synthesis, we have so remember chloramphenicol and also tetracyclines. So 5 antibiotics that I require you to remember only. And for the question number 7, so for these questions, we have 3 sexual reproduction in bacteria. We have a transductions when one virus can carry genes of the bacteria and transfer into another bacteria. Conjugation, this means that they transfer the gene through the plasmid through the pili rice. One have the positive containing the gene, we call them the donor, and one bacteria don't have the size of plasmids, we have the recipients, and they will transfer that gene through the conjugations. And the third process, we have transformations when some dates bacteria will release the gene into the environment, and some kinds of other bacteria can uptake this gene from the environment into their bodies. And for the transcription, the right is correct answer for these questions, because the transcription just only the process inside a cell by which the DNAs will transfer this information into the mRNAs, right? Or in RNAs, not really, or mRNA only. And the question number 8. Who can explain? Question number 8.
Well, it's the insulin produced by recombination DNA is identical to that of human.
Yes, sure. They are identical to humans because we take the genes and codes for the insulins from humans, and then we transfer into the plasmids of the bacteria, and that kinds of or plasmid contain the DNA from gene from the human. We call the recombinant DNAs, and then we take these kinds of recombinants or DNAs and we insert back into the bacteria, and these we produce the insulin that's identical to our human insulins. They cheaper and they have the same or effective. These are equal to the human insulin. Is no more effective. And the last question. Sorry, this This question is so easy, so there is no more explanation. The characteristic of the plasmid, they are extra genetic materials. Okay, so we have a big genetic material, the main genetic materials of the bacteria in the center, and we call them nucleolide. And they have another extra genetic materials we call the plasmids, and they are circular, they are naked, and no wrapped. They are naked, which means that they are no wrapped by the protein histones. So this all the question for the quiz today. So for the first 10 lessons, we study about the cells, either both prokaryotes and eukaryotic cells, the structures and the function that or its part inside the cells and the mechanisms under our mechanisms and how the cell can live, how the cell can communicate with each others, how they can divide, how they can grow. And for the last lesson, today is our last lesson, I will show you how we can culture cells out of living organisms. So how we can take our liver cell, kidney cells, or the cell inside our bodies and cultures in the laboratories, and how we can culture the virus, how we can culture the bacteria, and also the plant cells. And the lesson today is only more than 1 hour, so I think that we can we study now, and we don't have a break, and maybe we will finish 10 or 5 minutes sooner. Okay, this okay for you? You still need a break first. Sorry, you still need some minutes of break. I have no idea. Okay. Or maybe it means that you want a break. It's okay if we will skip the breaks. So, the last part, cell cultures and the applications of the cell cultures. So, when you read the medical studies, or when you read some article related to the medicals, or have you ever read? Have you probably seen that some of them are in vitro and some of them are in vivo? Do you understand which or the meaning of these terms? What is the in vitro and what is the in vivos? Do you familiar with these words? Yes, if we study straight, we will finish 5 or 10 minutes earlier. Do you agree? Because the lesson today is just more than 1 hour only. So come back to the lectures. So have you read any medical studies, and do you know the in vitro and in vivo, what difference between them? These two terms sound very similar. Guys, you don't look in English at all.
I think in vitro means in the test tube, and in vivo means in a living organism.
Yeah, sure. I agree with you. The in vivo is in the test tube. So when you see some couples, they have a baby by the IVF method. It means the in vitro fertilizations, thụ tinh trong ống nghiệm, right? So for the in vivos, it means the medical experiments are studies that's performed only in a laboratory or in a test tube, whereas the in vivos, they describe the medical experiments or a test that is performed only in living organisms. So the lesson today, we study cell cultures. This means that we studies the in vitro methods. So these two terms sounds familiar, but they are the big difference between them. Even summer papers is often gets confused with these two terms, right? So we study cell cultures, we study the in vitro cultures. So what is the cell cultures? It is the process by which the prokaryotes, eukaryotics, or plant cells are grown under controls or conditions, or we call them the artificial environment. Yes, that we culture the cell out of our body, so we need to create the artificial environments for them. So the first artificial environments were discovered is the ringer solutions. In the 19th centuries, we have these scientists named Sidney Ringer. He developed the Ringer solutions, and they contain several salt dissolved in waters for the purpose of creating an isotonic solution, and they are familiar to the body fluid of an animal. So the ringers solution, they contain some salt like sodium chloride, potassium chloride, calcium chloride, sodium bicarbonate, and balance the pH. The familiar like the fluid, the body fluid of animals or humans. And in 1885, the Roots removed the section of the medullary plate of an embryos of chickens, and they can counter it in the worm's saline solution for 13 days. And this is the first time that we proved that the tissues could live outside of the bodies. Medullary plate là tấm tụy của cái mô gà. And during the histories in the Harrisons, the first persons who successfully grows artificial tissue cultures. But so in the past, so now we have we get a serious problem with the COVID-19, right? So so in the past, we the mankind used to have the big problems with one kind of the virus we call the poliovirus. And they call the polio or poliomyelitis disease. So do you know this kind of disease? Bệnh bại liệt ạ?
Chứ không phải uốn ván hả cô?
No. And for this disease, you know this kind of disease. So this will kill you by suffocation. Bệnh ngạt thở ạ. So it's not damaging the lungs as the COVID-19 does, but it attacks the motor neurons in the spinal cord, and they weaken the communication between the central nervous system and your muscles. And then the muscle have no enduring paralysis, and they make it possible to breathe no longer work, and the patients will be dying by that they cannot breathe and they will die. So when you surf the internet, have you ever read about the man with the iron lungs? So because the patients cannot breathe, so the human that develops one thing, we call them the iron lungs. Some machine like this. Have you read about the iron the man with the iron lung, the Paul Alexander? He's now is more than 70 years old, I think. And he's the last people in the world still using these iron lungs because he's caused the polio virus in his childhood. So there's still serious problems. It's just because before in 1930s, we still culture the virus in the living monkeys. So when we want to extract the compounds from the living monkeys is take times, it costs monies, and also they have series of side effects. And in 1954, the first Nobel Prize in physiologies or medicines were awarded to three scientists, John Andrew, Thomas Wells, and Frederick Robbins, because they were sorry, because the polio vaccines were first available because they discovers how we can culture the tissues outside of the bodies. They can culture the polio virus on the tissues, so they get the Nobel Prize. And by this, there's inventions, by there's discoveries in 1955. The first polio vaccines were available in 1955. So then until 1970s, the US has been polio free. And around 1970s, the development of mammalian cells culture techniques is there is developed the mammalian cell culture techniques. So nowadays, the cell cultures, the mammalian cell culture techniques is widely used in laboratories. So we can develop the vaccines, we research about the vaccines, we can perform the research about the cancers, and also protein therapeutics, etc. So there's numerous of experiments we can perform when we're using the mammalian cells culture techniques. So how we can culture the mammalian cells or the animal cell cultures. So the first part is the animal cell cultures. There are two categories of the cells when we culture in the laboratory. The first is the primary cells, and the second is the established cell lines. So we will go to discover what the difference between them. For the primary cells, they are obtained directly from the donor tissues. So there is no transforming those immortalized cell directly from the tissues, and they're only able to divide in a finite number of times. And remember about the telomere. When the cell keep divides by number of time, they will be dying because they lost the telomeres, and there is a process we call the senescence. Cells it grows under the right condition. Primary cell can grow and polarize, but by a number of times. And even Earth, when we culture this mammalian cell in the lab, like we take I take some bone marrow cell from the mouse born for members. I culture it for only 1 week to perform my experiment, and then I threw them away. So primary cells take directly from the tissues and limit numbers of times. And the second type of the cells, or when we culture the cell, we have established cells. So where the primary cells are obtained directly from the donor tissues, the cell lines can be delivered from the clinical tumors, or they are able to escape from the normal constraints of the cell cycles, and they grow indefinitely time, so we can call them the immortal cells. So we can use them for a long-term research, and we can obtain from the clinical tumor, and also we can transform the primary cells with the viral oncogenes or chemical treatments. So back the information from the oncogenes in the cancer cell lectures. Okay, so we can transfer the viral oncogenes, or we can treatment with the chemicals. This reagent or treatment targets one of the checkpoints of the cell cycles, and these checkpoints there to ensure that the cell meet the appropriate requirements in order to move to the next stage of the making the daughter cells. So the cell will grow uncontrollably because they have invade the checkpoints, and then we have the established cell life. So here's we have an established cell life because we treated or we take from the tumor, and they can grow, they can cultures in the lab, and we can use for long-term research. So in my laboratories is most of my cell life, they are established cell life. And here we have the first cell lines in the world. They are immortal cell lines, the HeLa cells. They are they were originally cultivated from the tumor cells of the patients who dies of the cervical cancer, ung thư cổ tử cung. So they take from the tumors of this cervical cancer. This one, cervical cancer. So they take this from the cervical tumors, and they isolate these kinds of the cell lines, HeLa cells, and these are still keep continuous cultures. We're using so they are more than 6 decades now, and more than 60 years. But the HeLa cells still use, they still alive, and they use for medical research, and they are widely used in many areas of research. And also in my laboratory, we have the cell line also. From 60 years ago, from a patient get problems with the cervical cancers. And now the HeLa cells still survives and used in so many laboratories in the world. That is one famous established cell line. Cells, when we want to culture the cells out of your bodies, we need to provide them the environment that they can survive, right? And when we culture the cells, we have first we have two categories of the cells, established and primaries, and the second, when we culture the cell, we have two types of cells. One is the adherent cells, because the cell is attached into the bottom of the culture equipment. And the second, we can culture in the suspension cultures, because the cells is free floating in the medium here. So one example of this suspension cultures when we culture the blood cell. The especially red blood cell, because they free floating in your bloodstream, right? So now when we culture them, they are free floating in the cell cultures mediums. But for another cell type, they are adherent cultures, because they will attack in the bottoms of the culture flask. So to maintain the cells out of our bodies, we need to provide them some condition for them to live. And can you guess what kinds of the or what the components of the culture media? Yeah, môi trường nuôi cấy, media is the môi trường nuôi cấy tế bào. So can you guess what thing we need to provide or for them?
Maybe nutrients and appropriate
Nutrients, okay.
respiratory substrate.
Nutrition in what?
Maybe respiratory substances such as oxygen and other
Oxygens,
chemicals.
other chemicals, or this kinds of chemicals. Also, okay, I agree. First, we need to provide food for them, right? We need to provide food for them. And we have one thing that we call the culture, the basic culture media. This is the basic thing. They have the amino acids, inorganic salt, vitamins also, but it's not enough. This is basic thing we can make it, and we need to add another things we take from the cows bovine serums, the feta bovine serums, FBS. So it's collected from a bovine fetus. And is more we supplies for the basics of the culture mediums by some kinds of macromolecules, lipids, and also the most important thing, that's growth factors. So remember about the lesson of cell communications. One cells, they will be dying, they will go kill themselves if they receive no growth factor from the environment, right? If the no signal from the environment are promote them, so okay, units keep alive, they will kill themselves. So this FBS provide the growth factors also. And here we keep from cows. But one problem is the FBS here, because we collect from the bovine fetus, there are a lot of uncertainties regards to the use of the FBS, because the scientists do not know the serum's exact composition, and there is always subjected to the lots of variants, and this in turn can affect the cell cultures. And another things we need to maintain is about the pH. So we need to keep pH from the 7.2 to 7.4, and we supply the carbon dioxide from 5%, and of course they take the oxygen from the environment. For the buffers, we have some common buffering system, like sodium bicarbonate is the same like your bodies, or we have the buffering system, HEPAS. So you can search for internet for these two kinds of the buffering system if you really care about cell cultures. And we add also some indicators, pH indicator, we add a phenol red. So in normal case, when the pH is okay, we have the red like this. But when the cell is overgrowth, or they were contaminated with some bacteria, the pH will change. pH will change in the median, will come orange or yellows. This work like this. And beside the nutrition, beside the balance of the pH, beside growth factors, beside anything, beside all the things, we need to keep the correct temperatures. So when we culture the human cells, we need to maintain is 37 degrees. When we culture the pig cell, we keep them in 40, more or less 40 degree. So it's for the pig cells. And if we culture the cold-blooded animals, we need to maintain the cell that is suitable for this cells can live. So it depends on the origins of this cell. And the most important things when we culture the cell out of the bodies, that is we keep the sterilized environment. The contamination is the big problem when we culture the cells. They will be infected by some agents like bacteria, the cell when we contaminate it with the bacteria is like this. So you can see your cells, the human cells, in the shape like this. And the black dot here, they are the bacteria. And also they will be affected by the fungi and sometimes the mycoplasma. And by these kinds of reasons, we can work under a laminar flow hood, a box like this, a culture box, laminar flow hood. So the fresh air will go in and out. So in the environment inside the hood like this, they are sterilized environments, so we can culture the cells. And also we can put into the cell culture medium some kinds of antibiotics to eliminate some kinds of common bacteria that could be contaminated our cells. And one thing pay attention is the cross contaminations. So it may leading to the false scientific conclusions. So for example, is my students, when I work in the HeLa cells, I also ask him to work with the HAP cells. The HAP G2 is from the liver. So sometimes you will make the problems in the cell cultures, do not change the tape or some other technical problem. They will make the HAP-G2s contaminated with the HeLa cells, and it's leading to the false scientific conclusion that the problems when you culture the cells. And so that's all about the animal cell cultures. What about the bacteria? So for the bacterial culture, remember my previous lesson, there is only less than 2% of bacteria can culture nowadays. Remember? Less than 2%. And for the archaeas, it's most of archaea, I think this almost, and maybe most of them, or all of them cannot be cultured, the archaeobacteria. But less than 2% of bacteria can culture nowadays. And most of them is come from our digest our systems, right? So we know the environment that them live, and we can culture like we can culture E. coli and some common kinds of bacteria. And remember from my previous lesson, is nowadays just more than 2,000 or bacterial strain can culture nowadays only. And most of them are in our intestines. So how we can culture bacteria? So this is a common way that we can culture the bacteria. We have some agar. This is mediums for them. We put nutrition also, and we put some medium in containing the agar, and so agar, and this make the agar place. Agar, so make this medium become solid medium. So in the first, we have two ways. In the first way, we put the bacteria, and then we put the mediums, and then we colonize with growth. And the second way, we prepare the peptides with the medium in the agar mediums, and then we use this. I forgot the names in English of this. Quay kỹ. So we use this stuff to squirts and spread all the solution that's containing the bacteria in the surface of this agar place, and we incubate in the number of times. So if the bacteria come from our bodies, we can incubate in 37 degrees, and it takes more than 1 day. All the colonies will grow. X colonize here this from one bacterium. So this is the one culture dish of the bacteria. Is look for like a picture like this. And the second ways that we can culture the bacteria like in the flask like this. in the flask. So in the liquid form, so that's why our liquid form. So it means we culture the bacteria in the suspensions. Imagine like you make the yogurt, right? You have your meals, and then you put some more parts from the the yogurts, and you put back in your meals. You incubate for 1 day, and then you have another type of yogurt that makes by yourselves. That's the way you culture the bacteriums. And in the laboratories, we have 2 ways. First, you a medium containing with the agar and make the solid surface to grow the bacteriums. And the second way is we put them in the flask like this, in a liquid medium. We incubate, and then we have another growth of bacteria. And that is about the bacteria. And how about the culture of the viruses? So from the previous lesson, I told you that the virus is not really a living organism, so they only can grow inside a living host cell, right? So how we can culture them? So first, when you want to culture the virus, you need to isolate the virus from the pathogens, from the patient. Like, so unlike the bacteria, many of which can grow on the artificial mediums. The virus require the living host cells, and the infected host cells can be cultured and grow, and then we can use them as the grow mediums, and then we can have the sources of the virus. So when we receive this source of virus, we need to isolate them. So they can separate by either centrifugations or filtration. For the centrifugation, is maybe it's familiar with you. We use the speed. And this virus is more lighter compared to the host cell, and then we can collect this part, the lighter part. And what about the filtrations? Here we use a for the filtrations we use the paper, the filter papers like this. So here, you know that the blue one is the animal cells, the red one the animal cells, the blue one is bacterial cells, and the green one is the virus. So when we use the filter papers with the pore size of 5 micrometers, we only prevent, so we collect only the bacterial cell and the virus. If the filter pore size less than 0.2 micrometer, we can collect the solution with containing only the virus. And here's our, so is magnified from here. Magnify from here. So this is pictures of the filters. The pore size here 0.2 micrometer. So you can see that some kinds of bacteria in the root shape are stuck there. So we can collect only the solution with the virus. And how we can culture the virus? We culture it in the host cells, and we need to culture the virus to identify and to diagnose the pathogens virus in clinical specimens. So when the COVID-19 pandemic happens, so first they need to isolate, they need to culture these kinds of the COVID-19 virus, right? And then we can identify, okay, they are the new types of virus that we have never met before, and we name it the COVID-19. And the second, we can culture them to produce some kinds of vaccines. And in general case is, in general research studies, we need to culture the virus to perform some kinds of projects relating to the virus. The virus can be cultured both in vitro and in vivo. So in vitro, it means we culture in some kinds of flask. This is a flask that we use to culture the animal cells. Oh. Animal cells, and here's this is the agar place when we culture the bacterial cell. So here after culture, the animal cell were infected by the virus, and then we can collect this kind of suspensions. We use the centrifugations, or we use the filters to take the liquids containing the virus. Or in the second case, we if the virus affected the bacteria, we can use the agar plates. Here you can see that the virus, the visuals virus, the clone, we cannot see the number of virus, but here we have a number of the clones of the bacterium here. In the next step, some of them will be lysed by the bacteria and by the virus. And in the place number 3, you can see that's almost of the bacteria has been lysed by the virus. And then we can collect these, we can separate, and we can take the virus for our purpose, either the diagnosis or production of vaccines, or we perform some research. And this is about the in vitro. We culture the host cells or infected with the virus, and we can multiply of the virus. In the in vivo, also we can culture the virus in the hen's eggs. So you know that most of the influenza vaccines were manufactured for a new flu vaccination program is cultured in the hen's eggs. So you know that the eggs is not for the breakfast only. You know, they are the source of your common flu shot. Okay, so by which somehow people allergy with the eggs or the hen eggs, they refuse the influenza vaccines because there's may some kinds of possibility that they will have the allergy with that kind of vaccine if the purification is not work well. So what about the vaccine of coronavirus? Can you use the eggs to counter the coronavirus? Do you have any information? Can we culture the coronavirus in the hen's eggs? So we culture the coronavirus here, and we can produce some kinds of COVID-19 vaccines from the hen eggs. Do you have any idea?
Maybe we cannot, because it's maybe different kind of cell, and it has different antigen.
Or different antigens. What do you mean when you talk about the antigens? So, for the influenzas, okay, and actually the COVID-19 is, they said that the structure is familiar with the virus that caused the flu. But when the coronavirus pandemic spread, scientists and some governments around the world really want to develop some vaccines, but the X won't be the answers. They are due to having some different receptors and other characteristics. The coronavirus is able to replicate inside an X. the ways through virus scans. And because of this, to solve the problem of the COVID-19, we are pushing for non-X alternative in recent year, non-X vaccines. Oh, the problem. So non-X alternative. Oh, the problem with my pen today. Sorry. So we have a non-X alternative. Thus, because of this, we have one very modern vaccines like the mRNA vaccine. Remember mRNA vaccines? We use the mRNA, we put them inside NLPs, lipid nanoparticles to live in our cells, to live inside our bodies. So we have first the mRNA vaccines. So it's the first. We have Pfizer and we have a Moderna, right? Have you taken the Moderna, Pfizer's or AstraZeneca? So in three ways, we in the second ways, we have some kind of vaccines like AstraZeneca. So for the AstraZeneca, we put also the genetic materials of the virus inside. Now it's not a lipid nanoparticles, but we put inside another retrovirus, another safe virus, not COVID-19, another safe virus, because first it's safe, it's not causing the disease in humans, and the second way is easy to multiply them inside the living host cells or in eggs or cells. But it non-X anti-native. And in the third way, you know what? We have AstraZeneca, is some kind of Sputnik. In the second ways, we produce the virus. And the third ways of the non-X alternative. Can you classify, can you tell me another type of vaccine that you know? COVID-19 vaccine. We classify in the three types, right? The first is, so in Vietnam, we have a job that we call the Pfizer is the vaccine grandfather, vaccine ông ngoại. And the second type, we have some kinds of AstraZeneca. And the third,
Sputnik.
or Sputnik is much more familiar with the AstraZeneca. It's the same way.
Sinopharm.
Yes, the vaccine from China. And the third ways, we actually the COVID-19 cannot culture in the hands X, but they can culture it in fact one cell that can express the protein we call the ACE2. So forget for that proteins. The COVID-19 virus can grows inside one kind of cell. It is the kidneys epithelial cell for monkeys. So we call them the viral cells. We culture the virus, but not in eggs. In the viral cells, it's the kidney cell for monkeys. So that's the second ways we can produce this kind of vaccines. So we have a viral cell, we have a Sinopharm, that's two types of the vaccine from China. Is the third way. Okay, that's all about the culture, the animal cells, bacterial cells, and the virus. This in this graph, show you some applications to culture these kinds of cells, Plus, we can use the motor system to avoid uses of animal for preliminary studies. Of course, in the past, when they want to test some kinds of drugs, we need to test first on the animals, so even humans. So nowadays, by the ethic in science, so we refuse. There's no first treatment in humans. With the first treatment that we can use in vitro, that we tested in the cells, test in the tissues, so it's safe. We started to test in the animals, and by last stage, when everything is safe enough, we have the clinical trials in humans, in volunteers. But now, to use the in vitros, we can minimize the numbers of the animals in our research. So another types of applications like testing the effect of the drug on the cell growth. So is in my laboratories, we test this. We have numbers of drug form to test, and where to test in, especially in the cancer cells. We test also drugs on the cancer cell, like here. This used for the tissue engineering. We transfer some genes into the tissue and to see what happened with the genes, and also production of the virus, vaccine, and the antibodies. Study the virus, study the embryos, produce of some therapeutic protein. So numbers of applications in so the fields of medicine, I think, that first work in the cell cultures. Cells are in the second part. We go to the plant tissue cultures. I think it's another field of the cultures. For the plant tissue cultures, so the same as the animals or bacteria, the first we need to provide the suitable environments for them to live. Like first we need to produce or provide them the nutrition and or antiseptic condition that avoid any contaminate, and aeration of the tissues is mean here. Here's one types of plants can be regenerated from the single cells, with the apical meristem. So meristem here, meristem. Mô phân sinh ạ. We take the apical meristems of the plants, and then we put in the culture dish, antiseptics, provide nutrition, and in this the agar place is solid to make the mediums for the plant cells is solid mediums. And from that we put some kind of the growth hormones, numbers of the growth hormone to make them grow into complete plants. So that's why also is the same characteristic like the stem cell, right? So from one cell or some one small part or some from one tissue, we can regenerate into the completed body. So this kind, because the meristem cells in a plant cells, they have the characteristic like the stem cell, that totipotencies and plasticity. So the totipotencies is the ability of the cells can generate the whole bodies from the single cells or from the tissues by continuously division, right? And only in plant cell, only the meristem cells have that abilities. So we have this xylem vessels, they are the dead cell. They are, so you cannot cut the part from the stem here to regenerate into a completed plant cell. It's too difficult. Most of the totipotent cell abilities is come from the meristem, and also the plasticity that they allows one type of tissue or organ can form another type under influence of chemicals. So the application of the plant tissue culture does is micropropagation. Micropropagation does mean you can make the whole plant cell from the small piece of the plant, and is the advantage that the high rate we can produce the disease-free plants, and it's cheap. It's very cheap. So here is one micropropagation process that I take the staminate from flower. Here, we grow it into mediums, and we wait some time to have they can form the callus cell. They form the callus here. They form the callus, and then from the callus we put some kinds of the growth hormones, and they will form the embryo. And we take these kinds of embryo, we put in the environments, and we put some kinds of growth hormones to grow into completed plants. Like here, we put more alcins. So alcins to initiate the root formations, and then cytokinins to lead to the shoot formations, etc. Here's micropropagation is one type of one application of the plant tissue. We can produce numbers of plants with characteristics that we want in very fast ways, and really cheap compared to the traditional ways. And the next applications, last we can take advantage from some parts of plant that we can take some kinds of secondary metabolite productions. Here, thus we have some path we call the hairy root. Hairy roots, actually they are transformed root cultures. Các rễ được biến đổi hoặc là được chuyển các gen mong muốn. Hairy root cell cultures. Cells, we can produce some kinds of compound ones under the controlled conditions, and the quality of products will be consistent. They are the same, and we can produce also the novel compounds, and it takes less time and it's cheaper compared to, like, imagine that if you want to take some potential compounds from carrots, for example. So in traditional ways, we need to culture the carrots, so million of the meter squares of the carrots too, and then you need to extract this compound. So it really take time. It really expensive. But now you can transfer that unwanted genes into the root, produce the hairy root, and then you can culture only that kind of root, and you extract the compound from the root only. Is it's cheaper, and you take less of the times. So for example, you can we have some kinds of compound that we take from the culture the hairy root, like quinines. We can take from culture the hairy root, zinsenoside is one very important compound from ginsengs. We can culture the root from the ginseng to take the ginsenoside, etc. And another applications when you can with the plant cell cultures, that is one thing that's still debatable between the pros and cons of the GMOs. Here's I will introduce you some steps. That's how we can produce a GMOs. So there is one kind of bacteria living in the soil, Agrobacterium tumefaciens. They have the plasmid also. It's very beneficial for the genetics engineering, the plasmids. We take the plasmids from the Agrobacterium tumefaciens. We cut them to open it. the another type of DNA, another type of genes will be transferred into the plasmid. So the same like we use the to make the insulin, right? We take another gene into the plasmid, and then we transfer back into this kind of bacterium. And this kind of bacterium will infect some tissues, plant tissues in laboratories. They were infected with the plants when we do the tissue cultures, plant tissue cultures. And the affected tissues will be grow, or like we put some kinds of hormones, it is grows and this will become the completed plant. And now this plant is GMO, genetically modified organism, because in the whole part of the plant they have foreign genes here. So do you think that the GMO is benefits to the mankind, or you are the ones who don't want to use the product from the GMO? Is this okay for you if you use some product from GMOs?
Well, as long as they don't produce malfunction proteins, then I think I'm okay with GMOs.
Do you think it's good or bad? You think GMO is good for human or not?
I think it's neutral.
I mean, it solves world hunger problems.
Yes.
And also it provides, I guess, some kind of supplement inside the food, so we don't have to buy extra supplements. And one example of that would be golden rice.
Yes, the golden rice. So it's for African rice.
Well, yes, and it provide vitamin A.
Yes, and to curb the eyes of so many children, right? But why are people it's just are fight against GMO? They don't want to use the GMO anymore. So the big wild GMOs that is to curb so many life in the world. So if we don't have GMOs, maybe humans has been died by because by starvation, by hungers. They don't have a right to choose GMOs or non-GMO product, right? The huge benefits to the mankind, but why we still, or a lot of people still do not accept the GMO? So if in some product that they label with this or GMOs, this product is made from the GMOs materials, or in another type of product that is non-GMOs or GMO-free, so which product will you choose?
I think most people would choose the organic ones than GMO. I think it's just a stigma because people just are afraid of anything that is modified in terms of genetic level, at a genetically level. That's why they are afraid. They don't maybe they don't fully know what's the benefits of GMO. They just see the modified, and they are already afraid of the product.
Or some kinds from stranger things. So you take the gene from the stranger stranger plants to insert in your plants. But for me, for example, here I show you an example. This is the micropropagation. I made it. So this paper, this picture is come from my master thesis. So in my master thesis, I introduced GMOs, but in cacao trees. So it's a project or a collaboration between Vietnam, US, and Brazil. So Brazil is very strong in the cacao trees. So in this project, I take the gene from cacao tree, from this tree, and I put them back into the same tree. So it means that gene here is come from that tree also. It's not different gene. And I just wonder why the humans still afraid with the GMO. This is some kinds of modification. This not come from the stranger genes. We do not put the stranger genes into that kinds of plants. We put the same gene from the plant. I isolate the kinase genes from caca tree to perform the recombinant or TI plasmid, and then I put back into the caca tree. So you think that's still the problems?
Well, no, normally it only comes from misinformation. People that think that the modifications are bad from fake news and not really from people being scared. They just don't have the right information, so they draw illogical and unreasonable conclusions.
But then,
Is okay. Sorry.
oh, pardon me, but I think people just fear what they don't understand.
Actually, even they understand, but we need time to evaluate. We need time to prove that there is bad thing happens or everything is safe. So until all the information is not clear, so I think some people is that choose because they afraid about the GMOs. But in some countries like the Africans, maybe they are for the GMOs is their saviors, right? So GMO is still debatable in the world right now. And okay, now I will send you the links for the attendance checks in the chat box. Okay. This is our last lessons, and our series in tutorial class. We have 3 hours tutorial class, and if you have any question, prepare to ask me in this class. Okay, see you next week or the weeks after. Sorry. So today is our lab lessons. So if you have any comments, please let me know. Okay.
Teacher, when will we receive our marks? A point for the midterm test.
No, I'm not sure. Maybe next week. Okay. After your New Year vacations. 1 minute for advertising. So I have a laboratories and we're working in the project or we will find out the new drugs or new bioactive compound to treat some kind of disease like inflammations, cancers, osteoporosis, and numbers of disease in floor 2 in 205 in the room 205, and every years I will receive or 1 to maximum. Sorry. Oh, sorry, there problem with my battery. So I will accept 1 to 2 students to perform the research in in the field that's finding the drug to treatment some kind of disease. And the work is about culture the cells and to test the drugs and to find out the mechanisms underlying how these kinds of drugs can cause this kind of disease. So if you already care about this, can consider come to my lab. Just to avoid the contamination, in 1 year, I just received maximum 2 students only.
Then how can we apply for a position in your team?
Actually, in the second years, you have the group projects, and in your third year, for the group projects is from 3 to 5 students, and we work in one project we call the group projects. And on your third year, you will have 6 months to perform your thesis. That's you working in the research field. Also, for this is only for the students in PMAF, in MSD or FSD only. For the FSD, our student will work in some kinds of the oral plants, so we can produce some kinds of functional food. And for the PMAF, it's okay, so it's easier for me.
So if we want to be in your project, so what skills and knowledge should we prepare?
I consider the students like papers, and I will guide them from beginnings.
Yeah, thank you.
So, if you finish the attendance checks, you will leave right now. So see you in tutorial class if you want to see me again, because in tutorial class we don't have attendance checks. Remember.
Can I ask you one more question?
Sure.
If I want to apply for your team, then I must send an email to you, right?
Think about it in your second years.
Okay.
For the first years, normally you have a lot of subjects to study, so it's not okay for the first. But in your second year, you can send me emails or you come to see me in the lab 205. Okay.
Thank you.
Sorry, teacher, I just lost my internet connection. Can you resend the attendance check?
You can stay here. Attendance checks. Receipts. Các bạn muốn tìm muốn làm nghiên cứu về tìm hiểu các hợp chất thiên nhiên hoặc là tìm ra các loại thuốc để chữa các bệnh như là ung thư này. Hiện tại thì cô đang làm ung thư, làm các bệnh kháng viêm này và các bệnh chống loãng xương chẳng hạn. Thì đấy là cái đề tài mà cô đang làm, và nó đều thực hiện trên các tế bào, dòng tế bào động vật, các tế bào từ chuột hoặc là từ người, các tế bào ung thư ấy, nuôi cấy tế bào và thử thuốc. Đó, nếu mà bạn nào có quan tâm thì năm 2 thì có thể làm group project với cô, và năm 3 có thể làm thesis. Và cái phòng nuôi cái tế bào động vật thì chắc chắn là phải cần sạch tuyệt đối, cho nên là mỗi năm cô chỉ nhận tối đa 2 bạn thôi, không dám nhận thêm. Nhận nhiều quá nhiễm chéo lung tung hết cả phòng.
Mấy thằng mình không vào được.